Welcome to the Next Human. My name's Arav. Today I'm joined by Maria Rottersman. She's a PhD student at the University of California, Davis, in plant biology. So first of all, thank you so much for being here today. Um, and yeah, would you like to, tell us a little bit about yourself, your story, like yeah.
Thanks for having me. So my name is Maria. As you introduced, I'm a PhD student in plant biology at UC Davis. I'm originally from the Chicago suburbs. Um, yeah, I grew up there. I did a lot of in addition to science, a lot of music and like performing artsy things growing up.
Nice.
That's always been a big part of my life. Um, And then, yeah, my mom's a math teacher and my dad's an epidemiologist. So they both kind of like helped me decide that I want to go into science.
Mhm.
Um, yeah, and I went to my undergrad at the University of Washington, where I also studied plant biology, though my research was more on ecology. I did some grass seed dispersal ecology. Um, as opposed to genetics, which I'm into more now, which is something I think I see as more of a career path for me. Um, and I'll also mention that since I was old enough to work, I've been like working, and that was just a big part of my development and giving me a perspective outside of academia. And they weren't always science related jobs. I was working in food service and retail and childcare and things like that. So um, I just wanted to mention that because I think a lot of young people think that there's like a straight path and the other things are just distractions, but I think that they're really beneficial. At least for me, they were.
Yeah, I think that's a good point. And you mentioned that like you're like really into the arts and stuff. So I was wondering, did you ever like play any instruments, or were you like um or was it like mainly just um like physical art or
Yeah, I was mostly into performing arts. I did some theater growing up and I did a lot of band. I play the French horn. I still try to play when I can. There's a band on campus that meets once a week that I've been joining, and it's mostly undergrads, but it's just exciting for me to be there because it's like very unique to me, and I don't get to do it with my other colleagues in plant biology. So I'm having a lot of fun with that.
yeah,
And yeah, I did marching band all through high school and college, and yeah.
it's cool. But yeah, so how did you like first get into like plant biology, and how is that like kind of how did that, like you said your parents kind of helped you like realize that you wanted to like do science, but like plant biology specifically, like how did you end up there?
I will say that I kind of ended up more on the genetics mindset before plant biology. I was really into like fantasy novels and movies as a kid, and I told my grandma I wanted to make some like ridiculous horse lizard cross or something. And my grandma was like, "Oh, well, then you have to get really good at genetics." And that was a new word for me. I was like, I don't know, maybe 9 or 10. And I went to my school library and I looked up genetics, and there was one book, and it was supposed to be for teachers only, but nobody had ever checked it out. It was on plant genetics. So I read that and I thought it was the coolest thing ever. Um, got super into it. Um, and then in high school, we had a biotechnology class, which I realize is like sort of unique now. At the time, I
Yeah.
didn't realize that that was like kind of unusual for a high school to have that class, but I thought that was so cool and just learning about the up and coming technologies. And there was a section in the class on agriculture. And I just thought that was really cool and applicable and something I got really amped about. Um, yeah, then I went to college I ended up doing plant biology, but I was kind of on the fence between like molecular genetics and plant biology. And what kind of put me over the fence was this class called plant identification. And it was with Professor Dick Olmsted. And just like you look at a huge diversity of plants, and I think seeing all of that just really got me excited about plants. And so I already had this passion for genetics, and I have this passion for plants now. And now I'm in crop genetics, which I think is cool because there's a lot of applications. I'm very like goal driven, goal oriented. So being able to see that at the end of the tunnel, there's like something I can do for people and like something I can show people that helps them, I think is a huge driving factor for me and gives my work value and purpose.
That's great. But yeah, I mean, sorry, it's a little off topic, but like you're talking about genetics and stuff, and it's just like I'm sure you must also, but like wow, right? Like it's it really is just something like out of like a sci-fi movie. It's like I just can't believe like the stuff that people are doing and like you also in your work in crop genetics. And it's just sometimes I just like what the heck, like
Yeah, every once in a while you have those moments where you're like, wow, science is so cool.
yeah. But no, you mentioned that you're doing crop genetics, and like do you want to like kind of elaborate on the work you're doing and the people that it's helping? Because I think it's really cool stuff, and I'm sure you must also.
I do because I'm doing it. So essentially my PhD project is we are trying to make wheat that has reduced allergenicity or immunotoxicity for people with allergies or celiac disease or other sensitivities.
Mhm.
And so the way we're going about this is I work with a plant breeder, Wenjun Zhang, who has been breeding the plants to try to eliminate genes for the proteins that people respond to. And so what happens when a person responds to these proteins is gluten is like a huge complex of all sorts of different proteins. And even the proteins within gluten are like pretty big, pretty repetitive. But there's these tiny little peptide sequences that are recognized by antibodies. So essentially what we're trying to do is Wenjun is getting rid of those proteins in the wheat, and then I'm going to try to synthetically engineer the same proteins that'll hopefully still have the function of gluten that makes the bread and the pasta that we like, but without those toxic peptides that are recognized by those antibodies. And so in theory, if all goes well, we're going to have kind of like a gluten-free wheat where we reintroduce synthetic gluten that people aren't reacting to.
Nice. Um, so I'm not like fully up, like I don't know my statistics and everything, but like how much of the like general population do you think would like be helped or something by like this stuff? Because I mean like me, like my aunt is she has celiac, so like um obviously like gluten is a big no-no. So um yeah, like how many people, I think not just like people with this, but like also, so many people are trying to go gluten-free and stuff. My mom's cutting back and everything. Um, Especially with like food scarcity and insecurity being such a big deal. I mean, like how many, like, I don't know, percentage wise, how like big of like an impact on like the general population do you think this would like have?
Well, I will say that between 1 and 3% of the adult population has celiac. So
Wow, it's
it's
a lot
high
bigger than I
for
thought.
a
Like it,
disease.
like, yeah, wow. That,
Yeah, so it's pretty high. And then the other thing is that celiac has both environmental and genetic factors. So a lot of people are predisposed for celiac, um, and they won't develop it until later in their adulthood.
mm,
And I can't tell you definitively that eating more gluten brings out this celiac response. However, there has been correlative studies. And so in general, people think that if we're able to reduce the toxic peptides that your antibodies are reacting to, then maybe we can delay the onset of celiac, um, or even just have fewer or less extreme reactions. So even if we have the end goal is to create a perfect celiac safe wheat, but even in the meantime, we're getting rid of some of these peptides that people are reacting to, and we're hoping to at least reduce the allergenicity of wheat, and maybe that just reduces the number of reactions people are having or reduces the incidence of the disease in the population.
So I'm just curious, like how are like you like kind of like how did you get into it, or like how are you related to like the allergenicity of weed? Like what made you want to like, reduce it basically?
Hmm. So at UC Davis in the PhD program for plant biology, we do lab rotations. So
Mhm.
I actually did a few different rotations before I decided to join this lab and this project. But essentially, Jorge Dubkovsky, my principal investigator, he read my application to UC Davis, and my mom is from Turkmenistan, and he said, "Did you know that a lot of wheat diversity comes from Turkmenistan?" And I was like, "No, I had no idea." But then it kind of got me on this track thinking about wheat. Um, and I ended up just loving this project, loving how quickly you can see like the promising results of the project.
Mhm.
My rotation project was to take some of the wheat lines with deletions in the toxic protein genes,
Yeah,
and to evaluate the quality. So can we still make bread? Do we still have strong gluten matrices? Can we still, do all the things that normal flour would do? And for some of these proteins, it looks like the answer is yes, we can have the same or even better, stronger gluten, um, depending on which proteins we're deleting, and hopefully reducing allergenicity without compromising bread. That's like a huge step in the right direction.
that's great. Um, so yeah, like you said, sorry, you said your job was like, or your like assignment basically is like um evaluating like the compounds and seeing like is it still kind of like viable without this specific protein or something? Is that okay? That's cool.
That's a large part of it.
So like how big would you say like your whole like team or like lab is? Like how many people are like all working on this like with you?
Well, our lab is a lot bigger than the number of people working on this project. I would
Mhm.
say our lab is, I'm kind of guesstimating here, but maybe 25 people. And then on this project, it's primarily three of us, me, my PI, Jorge Dudkowski, and then Wenjin Zhang, the breeder.
Mhm.
But since we have this big lab, it's actually really nifty because I get to use other people as resources, especially as this is only I'm finishing up my first year. So there's lots for me to learn. There's individuals that are really great with stats and coding who are helping me with my statistical analyses. There's people who are really good with genomics, which I'm still learning about, people who are really good with more the biotech side. So I think a lot of people are chipping in, but primarily it's the three of us working on the project.
Nice. I think earlier you said you did your like undergrad at in Washington. Uh,
Mhm.
and yeah. So so you're saying is you just said you finished your first year. Did you just go straight from like your right after your bachelor's to this work, or did you like do something in between, or just kind of, yeah, like I'm just curious.
Yeah, so I started working for a biotech company as a lab aide while I was in undergrad.
Hmm.
And then after I finished undergrad, I got hired as a full-time research associate there. Um, and so I worked there for about a year before coming to grad school. A year, not counting the stuff I did in undergrad. So total maybe 2 years, but like 1 year full-time. And this company was in protein manufacturing and design. They're
Hmm.
called Zeta. They're in Seattle. They do really cool science. So essentially, my job was to help design the genes that we would then transform into E. coli, and then the E. coli would manufacture the protein for us based on what we wanted, and we'd evaluate, different versions of the protein, see what works the best.
Cool. Um, yeah, so you said there's like three of you like working on this project and stuff. And um, yeah, so you said, sorry, you said you're in your first year. So you obviously have like a lot more um years working on this ahead of you. Are you um are you excited?
Yeah, I'm definitely excited. Um, I think right now my up and coming project is to design these synthetic gluten proteins that we're going to introduce, which is kind of up my alley based on my previous job in biotech. But when I worked in biotech, I was working with E. coli, which has a much smaller genome that we didn't have to think about as much because there aren't like organs for you to worry about where things are being expressed. And then the wheat genome is just enormous, and you have to worry about when genes are going to be expressed and where they're going to be expressed. And it is a very different ballgame. So I'm excited to learn about that and to kind of apply my skills to this new system and this new project. I'll also say that I don't really know what I want to be when I grow up. So I'm just excited to explore. I do a lot of work with the California Wheat Commission, which is a USDA funded lab that they test the quality of wheat, which is what a lot of my first year was. So I want to get more involved with them and kind of like see science communication and how they interact with the community, and kind of decide where I land in the whole big world of crop science.
Nice. Yeah, I mean, and like Davis is like amazing for like everything agriculture related. So I mean that's amazing. But yeah, I mean, so do you guys like also like grow your own wheat like in the lab and stuff, or just or is like most of the work you're doing like like theoretical or like on the you know computers and stuff, or
I think everything starts on the computer because you want to make sure you have something good and worth testing before you put it out there.
yeah.
But we do grow all of the wheat. We have a little, not little, we have a farm. It's called, well, I don't know if it's considered a farm because we're not like, selling or mass producing anything, but it's pretty close to campus. And we have some folks in our lab working there, and we're producing a few kilograms of seed each year for each of our trials for this particular project, which I think is really cool. And I also get to be out in the farm and see it actually happening. I will say that there are some regulations on if you're going to do transgenics or if you're going to do CRISPR,
Mhm.
where you can grow those things. So as we start introducing these synthetic glutenins and as we start refining our gene deletions with CRISPR, those things will not be grown out in the field. They'll be grown in a greenhouse. The regulations change, which they might. They have been, I think, becoming more liberal in favor of modified plants. So
Mhm.
we'll see. But in the meantime, the plan is to grow it in a greenhouse, which will be a challenge because it's such a small space.
Yeah, I mean, is it like something you guys are like worried about? I guess you wouldn't be worried about like regulation like changing, but it'd just be something you'd have to consider and just deal with.
I think in a perfect world, the regulations will change because there's a lot of pushback on using biotechnology to modify crops.
Mhm.
And I think a lot of that pushback is from the public, and then it's implemented kind of on a national or statewide scale. But I think as people become more knowledgeable about how they're produced and become more comfortable with them, if we're able to change those regulations, then that could be huge because as of right now, the product, the end goal that we'd be trying to produce, I don't know if we'd have any economically feasible way of giving it to people.
Yeah,
So yeah, I hope that the public perception and then the regulations kind of change. But for now, we're just working with what we've got.
it's cool. I mean, like, I guess like regulation always takes a while to like catch up after like the general public and stuff. So yeah, I guess you just like gotta wait it out. But where do you think most of the like fears about like, oh, they're modifying our crops and everything. I think it's
Yeah,
just like there's just like unfounded like panic for like no reason, or people just aren't educated enough, or they're just like not willing to like educate them enough on like these subjects. Like what do you think the whole kind of like scare is about?
sure. I think there's a few different things happening, and I don't necessarily agree with a lot of the arguments people have for pushback. I don't want to say they're unfounded. I don't want to dismiss them because I do want to understand why people are scared or why they would want to regulate crops in this way. But I think the majority of it is just not understanding exactly how this kind of science works.
Mhm.
And there's also, a huge push for organic farming and more sustainable farming. And I think a lot of folks see organic sustainable farming at odds with biotech and the scientific advancements we're making. And I don't think that's true. I think they can both be used. We can collaborate instead of compete. But yeah, I think as people understand more how these things work and that they're generally, it's the same technology we've been using for lots and lots of years. We're just the same idea. We're just using new technology, and we're becoming faster and more efficient. And I think some caution is not a bad thing, but in general, I think the science is showing that it's working great. Yeah, I have a family member who's super into organic farming, and I gave her this book about how biotech and organic farming aren't at odds with each other. It's actually by Pamela Ronald, who is also a professor at UC Davis.
Mm.
And she loved it, and she totally was on board with it once she read it. She's like, oh yeah, we can both work together. We don't have to do one thing or the other. And this is just a new way of doing what we've already been doing as a society for thousands of years in breeding and agriculture.
Yeah. I mean, so in the name of like kind of like dispelling some fears, do you want to like talk a little bit about like the techniques you guys are using for like genetic manipulation and stuff?
Yeah.
Yeah.
So I guess I'll kind of go like chronologically with what the technology has allowed. So in the past, for thousands of years since humans started doing agriculture and farming,
Mhm.
what was happening is we were crossing plants with each other to try to create the best plant. And so there's a source of natural genetic variation there, and then selection based on what humans want, what works for them. And that's great, and it's been working, but it takes a very, very long time. And this variation you're looking for might not exist in the population you have.
Mhm.
So then kind of the next step technologically was to induce some of these mutations ourselves. So there's a lot of ways to do that. Most of them are kind of like, at least up until recently, a lot of them were kind of big scale, like zapping the plants in the same way that like you don't want to get a sunburn because you could have genetic mutations because of the sunburn. We're kind of like using chemicals or using radiation to induce mutations in these plants, and then again breeding them and selecting them to try to find the traits and the genes that we want. And we're still using that. It's a great technique. Again, it requires a lot of time and effort and a lot of plant material, since you're kind of looking at a bunch of random mutations to see if what you're looking for exists. And then more recently, with the advent of CRISPR and some other adjacent genetic engineering technologies and tools, we are able to alter individual genes. Sometimes we're able to just mutate them in a way that they don't work anymore. Or sometimes we're able to introduce new genes or enhance the abilities of these genes. And then we do the same thing. We breed and we select for the plants that have the genes that we want.
Mhm.
So I think that kind of freaks people out a little bit because it's new. But what I will say is that it's a lot more targeted than what we've been using for thousands of years. So, a lot of what we're eating now is like it's been zapped with radiation or with chemicals way generations and generations ago. And so it's still carrying some residual gene deletions from that. Even in breeding, we lose genes that are beneficial or important, even just with normal conventional breeding. And this new technique kind of allows us to select for exactly what we want. There's no outside consequences that we know of in the rest of the genome. Obviously, we do testing for that. We see if there's any other issues once we induce these deletions and induce these mutations. but it's a lot more targeted. And I think because that's how it works, I think that is reason to believe that it's safer, and it's certainly faster and more efficient than what we've been using. So yeah, we've got some stuff to learn, but I think it's really promising, and I think that it's safe and we haven't had any issues with it. So as long as we keep up that positive track record and we can get the public to be on board with that, then there is no reason not to use it.
Yeah, that's great. But yeah, I mean, so before we, wrap things up, there's like one last thing I like to ask people that I'm interviewing. It's what's like one piece of advice you'd give to high schoolers who are, thinking about getting into this like a similar field, or, this Gen Z basically.
I really had to think about this. Because I feel like I think I'm technically Gen Z, and I feel like I was
Yeah.
making these decisions not too long ago. But I think one of the big things is to keep up to date with the science that's out there currently. Like what you're doing is fantastic, and I think that it's a great way to learn. There's also just like if you read Science Daily or
Mhm.
something else where the recent publications are being summed up, it's just a good way to keep track of what's happening, because what you learn, especially in high school, it's really important, it's really foundational, but it's also often hundreds or thousands of years old. um, so I think that's, for me, learning about the up and coming stuff is more exciting. And then when you find something that you get amped up about, then you follow that. You can pursue that. You can see what publications come out of that research in the future. The other thing I'll say is that everyone should be really scrutinous of what they read,
Hmm.
especially, sorry, my cat is like just something. um, Everyone should be scrutinous of what they read, regardless of whether it's a primary primary source, a primary journal article, or if it's been summed up in Science Daily or another, major news outlet. Because everybody's trying to get you to believe something, which isn't always a bad thing. Like when I apply for grants, I need to convince the people giving the grant that my research is important. And it's not malicious. It's just, I have this opinion, I'm trying to share it. But there's other less noble causes, I think, out there. People trying to get you to buy something, or people trying to get you to oppose something. So just read, when you read and when you see this media, think like, what makes sense? What happens when I apply like critical thought and critical questions to what I'm reading or seeing? And where does the argument kind of fall through? And making those opinions and those judgment calls for yourself, I think it's a learned skill, but it's really important, because nothing you read is going to be completely taking all sides into account. Everybody has something they're trying to prove. So
Mhm.
just using your own critical lens to look at all of that.
That's great. Yeah, thank you for that piece of advice, and thank you again for agreeing to be here today. And yeah, I mean, that's it for today. So thank you.
Yeah, thank you for having me.